the introduction part, where this is the technical part of the biosensor. It has a kind of the receptor. The receptors are made up of either enzymes or the antibodies or the oligonucleotides, which are immobilized onto the surface of the bioreceptor, which we call it as a bioreceptor. And the biological event which happens between the target molecule, maybe for example the analyte, for example glucose, which we can see here, the glucose which will be recognized by the glucose oxidase, that is immobilized on the surface of the bioreceptor, and the enzyme reaction will be transduced, converted into other signal, and will be visualized either in the form of the graphical representation or in the form of the uh, kind of a digital display. So moving on to the other slide. So are the slides moving now?
Yes, yes.
Yes.
Yes,
So
Saitama.
this is the target molecule and this is the recognition unit. The recognition unit in the biosensor will recognize the target and the signal will be captured, will be transformed by the transducer, and the signal will be visualized. And these are some of the equipment where the biosensor is being kept into the market for the ready use. Coming to the principle of the biosensor operation, here you can see the surface of the bioreceptor. The bioreceptor is nothing but the antibody or the enzyme or the DNA, or in this case aptamers, for example DNA or the RNA aptamers or whole cell. These are all will act as the bioreceptors and the analyte in which we are interested. So this kind of the analyte and the bioreceptor interaction will be called as the biorecognition, and in the transducer that will be amplified and then processed and will be displayed in the form of the signal. So moving on to the classification of the biosensors. Biosensors are classified based on various components which are present within the biosensor. Am I clear in my voice and
Yes,
audio?
Saitama.
Yes,
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 Slides.
Saitama.
yes.
Yes.
Yeah.
Yes,
Okay.
Saitama.
Based on the bioreceptor, that is the first fundamental part of the biosensor. Here you can see this is the bioreceptor. Here the antibodies are immobilized. That is the meaning of the bioreceptor in the biosensor. Biosensor basically will have bioreceptor, that is the one which could be an antibody, which could be an enzyme, which could be an aptamer, which could be a whole cell, or which could be an organism itself, like microorganism. So that is called as the bioreceptor. Usually it will be immobilized onto the surface of the biosensor. So which we are talking about the bioreceptor. Based on the type of the bioreceptor, the bioreceptors of enzyme type means the biosensor is classified as the enzyme based biosensor. If the bioreceptor is an antibody, it is an antibody based biosensor. Aptamers, either RNA or the DNA oligonucleotides. Based on that, it can be called as the aptamer based biosensors, or whole cells are nanobiosensors. If the nanoparticles are used for the immobilization of various biological particles, then you call it as a nanobiosensors. So that is one type of the classification based on the bioreceptors. Because in the biosensor, the basic components, one component is the bioreceptor, other component is the transducer. Based on the type of the transducer, how the signal of the analyte and the bioreceptor will be turned. Either it is an electrical signal or in a heat signal or in a sound signal. Based on that, the transducers are based on the transducer type, the biosensors are classified. They are either electrochemical biosensors. In that, again, a category of the biosensors are there based on the type of the signal which will be generated between the analyte and the bioreceptor. There are amperometric biosensors or the potentiometric, which we will see in detail in the further slides. Voltammetric or the conductometric or impedometric. And if the signal is of the optical type, these are called as optical biosensors, or electronic biosensor, thermal biosensor, or gravimetric biosensor, or acoustic, if it is the waves type of the sound waves, acoustic biosensors. And if the classification is based on the detection system, These are called as the optical or electrical biosensor, electronic, thermal, magnetic, and mechanical biosensors. Based on technology, what type of technology you are using to make the bioreceptor. These are called as the nanobioreceptors or the surface plasmon resonance, SPR, or the biosensors on chip or electrometers. So these are the different types of the biosensors. For any biosensor making, we need to immobilize the biologically active component, otherwise we call it as a bioreceptor. So what are the methods here? Only for the enzymes it has been shown as the bioreceptor, but bioreceptors could be either DNA or the RNA in the form of the aptamers, or they could be antibodies, or they could be whole cells. So the mode of cross-linking or mode of immobilization of the bioreceptor would be more or less followed in the same way, either by adsorption or by the cross-linking or by entrapment or the covalent bonding or the encapsulation. Here only the enzyme has been shown as an example. In the semi-permeable membrane, for example, you can take as the lipozymes. Inside the liposome, the enzyme would be entrapped. So these are some of the methods, and in detail of the chemical part of the methodology I am not dealing. Am I okay in my
Yes, Sai Padma.
sound and explanation? Coming to
Yes.
the electrochemical biosensors, so these are basically used for detection of the redox enzymes, oxidation reduction enzymes, where there will be a generation of electrons or the abstraction of the electrons, either oxygen consumption. So this is just in a block diagram where the analyte, for example, glucose you can consider in this case. You can consider here biorecognition element or the bioreceptor as the glucose oxidase. The resulting event in the form of the electrons will be captured by the transducer, and the event will be uh, shown as the signal. So that is the basic understanding. Coming to the types within the electrochemical biosensors, one type are called as amperometric enzyme biosensors. Here the biosensor will give the signal due to the electrons flow. So in the form of the current. Why electron flow will be altered, which we can see. Again, there are three types in this. First generation biosensors. For example, glucose is there. Glucose gets oxidized here, where means glucose taking up the oxygen, but in turn it is getting dehydrogenated. That means H2 is going out from the glucose and resulting into gluconolactone. For the biology teachers, I hope I am clear. So during the production of the gluconolactone, whatever the H2 which would be produced will be combined with the bi-substrate. Oxygen will be used as the bi-substrate. So that H2 will combine with the oxygen, result in the form of the H2O2. This H2O2 will be and again gets converted into oxygen by releasing 2 electrons onto the transducer. So this immobilized enzyme, it is taking up 2 electrons from where? From glucose means gluconolactone. That means 2 electrons in the form of 2 hydrogen atoms are being lost from the substrate in resulting the product. So H2 is coming out from the glucose. H2 combines with oxygen, H2O2 at any point of time. The regeneration should happen for the next process of the enzyme. During that process, 2 electrons will be given to the transducer. So there is a change in the electron concentration, nothing but there is a change in the current. So that will be sensed by the amperometric biosensors. That is the first generation biosensors, and there are many examples which are given here for the first generation enzyme biosensors: glucose oxidase, glutamate oxidase, alcohol oxidase, lactate oxidase, lacase, tyrosinase. All these are working with the principle of the amperometric biosensors of the first generation type. And moving further, second generation biosensors, here the difference is there is a mediator. The mediator is in the reduced state. So here the substrate is taking up the electrons. Here we can see at the down, the substrate is taking up 2 electrons, in turn oxidizing the mediator. Mediator has to get back to its original state. So what it does, it takes up 2 electrons from the transducer. So there is a change in the electron concentration. So that will be detected in the form of the signal. So that is with respect of the second generation biosensor. Coming to the third generation biosensors, here the immobilized enzyme directly will take up the electrons from the transducer. So the transducer is losing the electrons. In turn, there is a change in the electricity. So that is with respect of the third generation amperometric biosensors. I hope I am clear what I am talking. Audience, one
Yes,
or
ma'am.
two.
Yes, ma'am.
Am I clear?
Yes,
Anybody in
Sai.
the audience?
Yes, ma'am.
Okay. So moving further, this is an example where the acetylcholinesterase is impregnated or immobilized on the paper. So this is an example which is given paper-based amperometric sensor. The enzyme is been means immobilized. Moving on to the other type of the potentiometric biosensors, here the change is detected due to release of the hydrogen ions, or there is a change in the hydrogen ion concentration. So here one example we can see substrates, sugars, or the amino acids, or the esters. The enzyme is immobilized on the transducer surface. The transducer will take up the hydrogen ions. So nothing but then a pH electrode. So the pH electrode, how you detect the pH due to the concentration of the H plus ions. So here also there is a release of the H plus ions due to the conversion of the substrate to product. That H plus will be detected by the pH meter. So here you can see the instrumentation. This is the working electrode on the right side. On the left side, it is the reference electrode. This working electrode will be having the immobilized enzyme membrane. So where the enzymes will be immobilized, either by using the cellophane or by using the nylon or the nitrocellulose membranes. The enzymes initially will be immobilized onto these membranes. For example, here is given the glucose oxidase, and that immobilized membrane will be very tightly adhesive onto the working electrode. So when the glucose oxidase performs its action, the resulting change in the H plus concentration will be detected by the potentiometric biosensor, and the potential difference will be reflected here. So there is an inlet for the pumping of the analyte solution. In the sample, if you want to detect whether the glucose oxidase is present or not, if it is present, what is the concentration of the enzyme. So this is a kind of enzyme assay, either qualitatively or quantitatively. Moving on to the other type of the biosensor, these are impedimetric biosensors. Here, the transducer will be firmly attached onto the electrode. Here you can see on the extreme left the electrode. Onto that the transducer will be bound. On the surface of the transducer, the biorecognition element. Here the antibodies are there, artificial binding protein, or DNA, RNA, aptamer is there, or the whole microorganism cells, as I told, bacteriophage also is been adhesed or immobilized. So the change in the means the antigen, if it binds to the antibody, the signal will be detected through this impedimetric biosensor. And if you see this C representation based on the concentration, even the signal also will be altered. So this is one more example for the impedimetric biosensor. Moving on to the other aspect, here till now we have seen the enzymes are bound onto the transducer or immobilized onto the transducer. But here the enzyme is not immobilized onto the transducer. Instead, it will act as a labeling agent. For example, one want to determine the concentration of the analyte. So the respective antibody will be immobilized initially onto the transducer. The analyte will be recognized by the primary antibody. The similar type of the primary antibody in free form will be made to bind to the target analyte. That primary antibody will be recognized by the respective secondary antibody which is coupled with the enzyme. So usually the HRP, horseradish peroxidase enzymes will be used, and by incubating this enzyme with the respective substrate which will produce the product, the product is in the reduced state. So it has a capacity to oxidize. So during the oxidation, it will lose 2 electrons. Those 2 electrons will be captured by the transducer, and again there is a change in the current that will be measured. So this is another way. Here the enzyme is not directly bound or immobilized. Instead, it is used as a label element. In the same way, here also, instead of the antibodies which are immobilized onto the transducer, here the antigen is immobilized, but the target here is concentration of the antigen, how much is there, which is immobilized onto the transducer. So target, the antigen which is known concentration is made immobilized. The target here is the antibody. If any antibodies which are specific for the antigen of our interest, they will go and bind. That is called as a target antibody. But we need to have a basic idea about the target antibody and use the secondary antibody which has the ability to bind to the primary antibody. The secondary antibody has the enzyme. Again, for example, HRP, when the enzyme is having its substrate, produces the product can be converted into the oxidizable product, that is releasing the 2 electrons onto the transducer. So again, it is a kind of the biosensor which is working on the differences of the electrons. Moving on to other type of the biosensor, this is called as a piezoelectric biosensor. So what is the meaning of the piezoelectric compound? These compounds as such, they are inert in nature. There are many chemical compounds. The best usable compound which acts as the peaceable compound or material in the biosensor is the quartz. As such, it is inert, but by having electrodes on both the sides and use force, this could be any pressure, or that could be any kind of the uh, mass difference, or the mass means weight. Weight means analyte. If any analyte is bound, there is a change here with respect of the mass, that also will act as a force which leads to the release of the current. That current on the where I am showing the cursor will be detected. So this is another example for setup of the piezoelectric biosensors. Here this is the quartz element. On both the sides there is a mechanical force, means in the form of the metals. Usually the metals will be put which will act as the electrodes because they have the capacity to conduct the current and use the force. By putting the force, electricity will be generated and that will be traveled. So in absence of the analyte, for example, antibodies, you are making them to immobilize on this kind of the transducer, on this kind of the biosensor. If the antibody is capturing any analyte, for example, antigen, there will be a change in the current. So that current change will be detected by the devices. So here you can see this kind of the difference in their voltage will be detected. I am not going in so much of depth about the piezoelectric components and the current paths because basically it is a hardcore physics. But what we have to understand, what I would like to convey my message is biosensor is one of the very upcoming field, and interdisciplinary approach for every subject is very much necessary because the physics person will not know what is the meaning of the glucose oxidase, but whatever the instrument which is being used is all for the medical purposes and all for the diagnostic purposes. So it is an interplay between the physics and the biology. So I would like to bring in that concept into my talk. That's why I have chosen this. And here there is an example given with respect of the antibodies bound with the analyte antigen. So earlier without analyte, here the red color, high frequency of oscillations happens on the piezoelectric material. For example, as I told you, quartz. But upon binding of the analyte, here maybe antigen you can take, the frequencies of these oscillations will be reduced. And here they have used even the nano particles labeled with the antibodies acting as the secondary means for the same antigen, but the antibodies are bound onto the nanomaterials. Again, there is a change in the frequency of oscillations. All these changes would be detected and they will be transduced, means transformed into detectable signal in the biosensor. Moving on to other type of the thermometric biosensor, are otherwise called as the thermal biosensors. Here, the difference of heat, because some of the enzymes, they are exothermic in nature, produces very minute quantities of heat. That heat can be detected by these biosensors. Even a small change in the heat generation will be detected. So these thermal biosensors are also of use. For example, one example is given here, serum cholesterol. This serum cholesterol, which gets oxidized by the enzyme cholesterol oxidase, releases heat that will be detected in this thermometric biosensor and calculated, and then you can correlate with how much amount of the cholesterol is there in the serum. So there are many examples like that for the determination of the glucose, urea, uric acid and the penicillin G, all were detected by using the thermometric biosensor. Coming to the other type of the biosensors, optical biosensors, they work on the principle of the electronic signal. So here there is a list. For example, surface plasma resonance and interferometers, resonators and gratings. So we'll just quickly move on. This is very important biosensor, surface plasma resonance biosensor. So the principle is like this. You can see here in a gold film, that is the electrical conducting material. So any polarized light, when it hits at the interface of two mediums, here on the right side, if you see, one medium is the glass medium, that is a prism, another medium is the liquid medium. So if any polarized light hits at the interface, there will be a reduced density, or the reduced uh, waves, density of the waves will be reduced. These waves are called as the plasmons. That's why it is called as a plasma resonance biosensor. Electrical charge density waves, otherwise. This polarized light, when it hits the electrically conducting material at the interface of two different materials, for example, here glass and the uh, aqueous medium, generates the very low density waves called as the plasmons. So these plasmons, they reduce the angle of the polarized light. They change the angle of the polarized light. So on the left side, if you see, this is the polarized light on the left side. The first diagram, if you see, without any antigen binding onto the antibody. For example, if we observe here, the black ones are the antibodies. There is no antigen binding. So the
केंद्रे मॅडम वर बसले मुलं जरा प्रॅक्टिकल पण करतात.
please
सुरू
please
करू का?
mute.
काय मुलं
You can check Rajni, that one. Who is talking?
Ma'am, you can continue. I'm muted then. Saipal ma'am, you can continue, ma'am.
Yeah. So when the polarized light hits at the electrical conducting material at the interface of two different material, the angle in this case is of different, but the same thing in presence of the analyte. For example, here what we thought antigens. When the antigens are bound to the antibodies, you can see the surface plasma resonance angle, this is called as a resonance angle, would be altered. So what is the altered angle? You can depict with the respect of concentration of the analyte. For example, here you can see this is the angle on the down, extreme down. This is one angle in absence of the analyte. And you can see here too, that means it indicates that there is an analyte which is present in the sample. That's why there is a change in the angle, resonance angle, surface plasma resonance angle. So here also in absence, there is no shift, but in presence of the analyte, the shift has happened, 1 and 2. So this reflected light changes its angle, that is called as a change of the resonance angle can be detected. And it is not only for the detection of the analyte, even which can be applied for the concentration determination. So this is an example where the fiber, after cleaning with the acetone and dipped in the silver nitrate leads to the silver nanoparticles, means they have dipped with the silver nanoparticle solution, making the silver nanoparticles adsorbed onto the surface of the film or of the transducer for nearly 30 minutes. And after getting adsorbed the nanoparticles onto the surface, and then dip in 3-amino propyl trithoxysilane, which results in the active amino groups. Why these active amino groups are necessary? For the linking of the enzyme, because you need the active groups, then only the what we have seen, the enzyme immobilizations, the enzyme gets immobilized. Then you use the substrate. What is the substrate here? Triglycerides. So if the triglycerides are there in the sample, they go and bind to this enzyme lipase. If the triglycerides are not there, then there will be a less shift of the angle. Here you can see, as the concentration of the triglycerides are increasing on the down graph, you can see there is a shift in the absorbance wavelength with respect of the nanometers. For every concentration increase, there is a shift in the wavelength. So this is a very good example where one will understand what is the concentration which is present in the sample with respect of the analyte. Moving further, there are so many biosensors which I have picked up. Coming to the just in nutshell, what we have seen, this is Amperometric. There will be a working electrode, reference electrode, and there is an auxiliary counter electrode. The electrons which are released will be detected in the form of the voltmeter, and this is nothing but the potentiometer, which we have understood. H plus will be more or less like in a pH meter, and this is a impedimetric biosensor, where the electrode is been attached onto the transducer, and the transducer surface, here the example is showing the nanoparticles which are having the biorecognition element. Am I audible?
Yes, ma'am. Yes, ma'am. Please continue,
Okay,
ma'am.
yeah. So like that, there are chemiluminescence biosensors. Everybody knows what is the principle of the chemiluminescence. Chemical compound releases light. For example, luciferin in presence of the ATP emits light. So this can be used as the biosensor. And luminol and H2O2, that combination also will work. So luciferase you tag onto any compound and use as an enzyme detection system, like as how HRP has been used in the earlier cases. Here luciferase can be used as the detection system. In the same way, luminol and H2O2 also will emit the light that is used in the biosensor, which is the chemiluminescence biosensor. Just now what we understood about the surface plasmon resonance, polarized light when it hits the electrical conducting material at the interface between the two materials, glass and liquid, the change of the angle can be detected, and here beautifully you can see the change of the angle in presence and in absence of the analyte. In the same way, there is another optical fiber biosensor, evanescent wave. So these are the waves which are generated with the help of the light source. If the analyte is present, for example, here again antigen can be taken here. The antigen is present, that will change the intensity or the oscillation frequency of the evanescent waves. So whether the analyte is present or not, one can detect based on the change in the evanescent waves, oscillations or the frequency. Moving on to the thermometric, already we have understood some of the enzymes will release heat during their reaction, which can be captured with the respect of the thermistor or the thermal biosensors. And here this is a nanowire based electron transistor. This is also is being widely used as the biosensor. And another surface acoustic waves, for example, waves or sound waves, all will be considered as the 1 minute. They will be considered as the acoustic waves. So if analyte is present, the acoustic wave oscillation and the frequency will be altered. So and here there are the material and in detail I will show in the next slide. There are two transducers which are called as the interdistal transducers, where I am putting the cursor here. This is one called as an input interdistal transducer. Here the uh, current will be generated, will be induced, and piezoelectric material which has the capacity to transmit the waves. So that material is at the base used as the transducer material, and to which the setup is like two interdistal transducers, one for input and one for output. So if the analytes are there, for example, the antibody is having a capture of the analyte, then the wave frequency or the oscillations will be altered. So that can be detected in the form of the output. Here there is an interdistal transducer, which is the whole setup is labeled as 7, and 2 is the piezoelectric material base. 1 is the solution in which the analyte is present, and these are the waves, acoustic waves, which travel through the piezoelectric material. Piezoelectric materials, there are many. I have skipped because of the lack of the time. Roselli, salt, quartz, and so many other piezoelectric materials are there to list. And this is a list of enzymes: glucose oxidase, electrode based biosensor. The principle is electrochemistry using glucose oxidation, and the applications like that, there is an uric acid biosensor, glycosylated hemoglobin biosensor, hydrogel based biosensor, silicon biosensor, quartz crystal biosensor. All there are so many biosensor people are working with. I have removed the references, and these are the biosensors which are used for the disease diagnosis or medical applications. For example, glucose oxidase electrode based biosensor or glycosylated hemoglobin biosensor. As everybody knows, it is useful for the diabetes. In the same way, uric acid biosensor is very much useful for the cardiovascular and general disease diagnosis. Nanomaterial based for therapeutic applications. Moving further, now the trend is moving on to the use of the nanomaterials. So using the nanomaterials, the respective biosensors, nanoparticle based biosensor, nano rods based biosensors, nanowires based, quantum dots based biosensor, carbon nanotubes, dendrimer based biosensors. There is so much of advancement is happening in the field of the biosensors. And here these are all the nanomaterials, either silver or the gold or the platinum, that is being used to make the nanoparticles. And what are they detecting? They are detecting aflatoxins and cholesterol H2O2, which is necessary for many of the reactions, and E. coli as such an organism, as a pathogenic organism for urinary tract infection sometimes, and for the pollution, or for the glucose, or for the metal detection, and adrenaline hormone, and urea, dopamine, hydrazine. There are many applications. I'm just moving very quickly. Glucose and then lactic acid, all these biological fluids which need to be detected will be very aptly detected by the biosensors. Now the field is moving on to the use of the field effector transistor biosensors. Here there are two components. One you can see labeled with the yellow and all. This is a biological recognition element, and other one is the field electro transistor. So when there is an antigen, the antibody is already immobilized on the transducer. And here we can see, based on the intensity of the IgG here, that is analyte, the signal also is varied. In presence of the 10 nanograms, in presence of the 70, 500, how the signal is being varied, one can imply that to the respective study. So this is a glimpse of what all the types of the biosensors which are in current use. The biosensors are being used even in the form of the contact lens. Nobody can even imagine that it can be used as a biosensor. And the patches, all the kind of the components, you need to have a biorecognition element, you need to have a transducer, all are embedded in this small patch. And here breath analyzers, which are very much widely used as a biosensors, and the biosensor is impregnated on the teeth to detect the microorganisms, or on the foot, R has the wrist patches to determine the diabetes flow or progress of the disease, or even the sweat is in a biological fluid where the electrolyte concentration, how a person is maintaining his electrolyte concentration, is being determined by taking the sweat as a sample for the biosensor. They will stick onto the body and it will be detected and given a real time indication what the person's electrolyte concentration in the sweat. So this is the whole setup on the wristband where all the components, which basically for any biosensor, one biorecognition unit should be there, and transducer which has the ability to convert the signal to measurable signal. So these two components are very important for the biosensor. Moving on to the advancements, because the title itself, past and present. Now the present work is happening on the graphene-based nanomaterials. Graphene has high advantages because it has large surface area and has a high electrical conductivity, high electron transfer rate, and it has the ability to immobilize different molecules. Here we can see there is an immobilization of the antibodies, DNA, and the enzymes. These all can be immobilized, so it is a best fit for the transducer surface. So this graphene sheet will be immobilized or will be made to attach to the transducer. So once the reaction happens between the antibody and the antigen, the signal will be converted by the transducer into a measurable signal. So all these molecules, these are the bioreceptors or biological recognition elements, you can call it as, which will be used to recognize either whole cells or the microorganisms or the ions or the molecules and even the nucleic acids. If you use the aptamer of single-stranded DNA, you can use that as for the detection of the nucleic acids in the sample. And the transducer material is made up of this graphene. And what are the types? As we have seen the classification, electrochemical, optical, thermal, and the piezoelectric type of the signal conduction happens at the transducer. And this is because it is an advanced field which is happening now, and enzymes can be immobilized, and here antibodies can be immobilized, DNA can be immobilized. Here one example is given on the graphene. The antibodies are there which will detect the virus, and then that is in a signal which will be transformed by the transducer. This graphene sheet is there on the surface of the transducer, which has the ability to convert, for example, electrons which are released will be converted into a readable record. In the same way here, enzyme biosensors as the graphene sheets, as the transducers, enzymes are immobilized here. Analytes, for example, glucose you can take in a simple example, the products will be released. Whatever the electrons are which are released, which will be transformed into a measurable signal. And what are the types of the enzymes mostly used in this kind of the graphene enzyme biosensors is lacase and HRP. There is so much of literature which is going on in this field. Coming to the use of the latest material, that is the 2D materials. What is the meaning of the 2D material? They are arranged in a single line. So this is the publication rate where the number of the articles are going year by year. Started in 2005 with a very low number of the publications. If we see in a span of 10 years, the number of the publications have been reported is reaching enormously to 1600 to 1800. And coming to the graphene and the nanocomposites makes up very high proportion out of the other 2D materials. So these biosensors, these are the materials, and in the next I have given what are the 2D materials. Graphene is one of the 2D material which is very widely used. Another materials are HBN, graphene, fluorographene, MOS2, WSCA2. So these are the materials which are arranged in the single layer. All the atoms are arranged in the single layer. So that is the meaning of the 2D material. And moving on to the application part, and wearable biosensor has very much wide applications, very easy to carry on with the wearable biosensors, still monitor in real time what is the patient status. These are all the pulse oximeter came to handy during our COVID time. This is the accelerometer which we use for our walking and so on and so on. And not only for the medical applications and the therapeutic, even for the drug discovery, what kind of the drug is suitable to study the drug interaction with the respective materials, carbon nanotubes and the graphene oxide. These are all the types of the materials which are used for the study of the drug discovery. These materials are in the form of the transducers. On the surface, biological material will be added, either antibodies or whole cell and enzymes. And coming to the applications of the biosensors, there is an list of applications: healthcare checking, metabolite measurement, for example, glucose, screening of sickness, whether the person is having a altered level or the normal levels of many parameters, for example, cholesterol or the lipids. Just now we have seen triglycerides, insulin treatment, clinical psychotherapy and diagnosis of disease in military, agriculture, veterinary, drug improvement, offense detection. Offense detection usually with the H2O to luminol, because they will get activated in presence of the blood, heme, iron. So in the offense detection, if any blood samples are there, the sensor can be used. It will detect. Processing and monitoring in industries, ecological pollution control. This is one big area where the biosensors are making their use in this field. Diagnostic and clinical, environmental, development of drug with using types of transducing material, and then process control in industries, pharma, manufacturing and all. So what is the market? It is increasing day by day. In Asia, the growth of the biosensor is much more than the developed countries due to widening health insurance and large population. And why it is necessary? Because the people are preferring from the hospital to home, because this regular monitoring is making their way of adoption very easy than going and then standing in the hospitals. So they can regularly monitor at their own place with very ease and comfort. So with this, I am closing my talk. I have taken all the resources from these references, and my humble submission, this is only an attempt to understand there is a requirement to integrate interdisciplinary